we left off talking about the properties of water before we didn't have time to do anything else here. So I know we covered this slide here. We'll just kind of recap it quickly here. We're talking about, a solution which has solutes in a solvent. If that solvent is water, it's an aqueous solution. And ions or molecules or anything with a charge or a partial charge are going to be able to dissolve in water. So we call them hydrophilic, meaning water-loving. All right, so this is where we kind of actually left off at. The example of dissolving table salt and how this actually works. All right, so we know that water is a partially charged molecule. All right, the oxygen has a partial negative charge. Hydrogen is partially positive. And because of these different polarities on the molecule, it's actually able to break apart any charged molecules that are dissolved in water. All right, so how it does this is basically, if we look at the salt here, so the salt molecule is sodium and chlorine here. All right, water is breaking it apart by literally separating the two apart. So the negatively charged oxygen ion is attracted to that positively charged sodium ion and kind of breaks it apart from the chlorine, where the opposite is true, where those positively charged hydrogen ions are attracted to those negatively charged chlorine ions. So it basically just splits that molecule apart and dissolves it in the water. Now this is true for pretty much any polar molecule that is going to be able to dissolve in water. The same basic concept is going to apply. Now there's many different types of molecules in terms of talking about them with water. All right, so we can have hydrophilic, which we kind of already touched on, water-loving. All right, they're going to readily dissolve in water because they have some sort of a charge associated with them. We also have hydrophobic, which is exactly the opposite, just means water-fearing. All right, these molecules, they don't dissolve in water very well, and they're going to be nonpolar. All right, so, such as hydrocarbons and oils, which we will actually hopefully get to later on today. Now we can also have molecules that have regions of both. All right, they can be water-loving and water-fearing. They are the amphipathic molecules. All right, so one thing about these amphipathic molecules is that they might form mycels. And detergent is an excellent example of a mycel. So on the next slide here, I have the actual structure of a mycel. And we can see that it has a polar and a nonpolar region. So these polar regions are on the outside, all right? So they are water-loving, and they are attracted to those water molecules. Now on the inside, we have those nonpolar regions. They don't want to be anywhere near water, all right? So they're all going to clump together, form this circle, and stay in the middle as far away from the water as they can. Now this is really useful when we talk about detergents because if we're doing a load of laundry, we want to get some oils off of our jeans or whatever. These mycels play a major part. So what it does is that it traps the nonpolar oils in the middle of this little mycel. All right, so it's trapped away from the water because oils, they don't like to be dissolved in water. They are hydrophobic. So they're going to go in the hydrophobic end of the mycel, whereas the polar regions, they're attracted to water. So they're going to be play an important role when we try to get rid of the oils off of the laundry. So doing the rinse cycle in the laundry machine here, these polar regions are going to be attracted to that water that's going to rinse off of your clothes. And because it is trapped in those oils in the nonpolar regions, it's going to take those oils or those pieces of dirt away with it. Does that make sense? All right, so mycel's pretty cool. We're going to briefly go over water in a few other aspects of water here. But because we kind of ran out of time last week, and I want to make sure we get to what we need to get to this week, I'm actually going to hand out a little worksheet for you guys to do. And I didn't put it on here, but I'll make it worth 3 points if you guys complete it and bring it next week. That's going to have you go over these aspects of water a little bit more in depth, do your own kind of research on that. But I am going to briefly touch on them here, just so you have an idea of what you're looking at. So we know that water exists in 3 different states of matter. All right, we've known this since we were in elementary school, solid, liquid, and gas. Now, changes in the state, especially in water, take a lot of energy to do. All right, so going from a liquid to a gas, going from ice to water, they all require an input or a release of energy. Now, generally, we can say the heat of vaporization is the energy to boil water, or the energy required to change water from liquid to a gas state. Likewise, we have the heat of fusion, which is the energy to melt the ice, so changing it from a solid to a liquid. All right, water uses a lot of energy to change states, and this is why it's so biologically important, is because of this ability to need a lot of energy. And we call this need, or I guess we don't call this need, we call it specific heat. All right, specific heat is kind of the term that's going to describe this need for energy. It's the amount of energy of heat to raise temperature 1 degree Celsius. All right, so water has a high specific heat. Think of how long you have to wait for water to boil on the stove, for instance. All right, it takes a lot of energy to raise the temperature of the water. Now, water, very extremely stable as a liquid due to the high heats of vaporization and fusion that we just talked about, and also because of this high specific heat. Now, because it is so stable and it takes so much energy to turn it into a gas that it stays as a liquid most of the time, organisms are going to use this to their advantage, all right? In fact, a lot of organisms, most organisms, have evolved to take advantage of water being in the liquid state and being very stable. Now, water isn't just a solvent. I know we kind of talked about it just as being a solvent so far, but they participate in many, many, many biological functions here. I just threw this slide up here just to give you kind of an overview. They can participate in chemical reactions, which we are actually going to get to today. Provide force or support. So think about if you haven't watered your houseplant in a while and it starts to wilt. Right? It doesn't have enough water to provide the structural support to keep it upright. It can remove toxic waste components such as the removal of toxins in the kidneys. Evaporative cooling is a very awesome one. So think about when you go to the pool on a hot day and you get wet and you come out of the pool and you're cold even though it might be hot out. All right, it's that water evaporating off of your skin and taking away the heat off of your body that's going to make you feel cool. So that's evaporative cooling in a nutshell. Cohesion and adhesion. All right, so it's the ability for water to stick to itself, which is cohesion, as well as adhesion, the ability for water to stick to other surfaces. So think about, water going through the veins of a plant, for instance. So it will stick to the sides of those plant cells as it goes up through the plant. We have surface tension, which we can see this little water bug here sitting on top of the water. Surface tension is very high in water because these hydrogen bonds in the water are very strong. They don't like to break, so they don't like to be exposed to air. So water is going to hold together as long as possible before it will give in. Hence the high surface tension. And finally, just as a lubricant, all right, you can see this dog salivating here. All right, so this is in anticipation of feeding, but it also can be a lubricant in like mucous membranes, for example, or keeping all the organs in your body bathed in some sort of solution so they don't dry out. All right, so very important as a lubricant. This is just a copy of what I just showed you guys here. So I mentioned before that when we talked about bonds, hydrogen bonds were the weakest of the bonds. That doesn't mean they're necessarily weak, they're just weaker than other bonds. All right, hydrogen bonds can be very strong. All right, and it's because of these hydrogen bonds that give water all these properties that we just mentioned, such as the high specific heat. It takes a lot of energy to break apart those hydrogen bonds between each individual water molecule. All right, so heat, the energy associated with molecular movement, temperature is a measure of that heat, and water has to absorb a lot of heat energy before those bonds are going to break. All right, hence the high specific heat. So those hydrogen bonds play a key role in all these properties of water. All right, any questions about the properties of water? I know I kind of went through it fast, but I'm going to have you guys do a worksheet to kind of solidify that information. All right, if there's no questions, we're going to move on to pH and kind of end this lecture here. We're really not going to go into depth about pH. I'm just going to give you guys a broad overview here. So in aqueous solutions, some water molecules can break apart, especially in the presence of other molecules. So if we release a molecule that's going to cause the release of hydrogen ions into a solution, we call that compound an acid. All right, we're releasing the hydrogen ions. Opposite is true about a base. All right, so if we're soaking up those hydrogen ions, that compound is going to be a base. Now the pH scale, I'm sure you guys have all heard of the pH scale here, describes how acidic or basic that solution is going to be. And especially in biological organisms, we all contain a sort of buffer that's going to help keep the pH in our bodies in an optimal range. All right, so that buffer is going to be some sort of a molecule that when hydrogen ions become too plenty in, say, the blood, for example, that molecule is going to accept those excess hydrogen ions. All right, try to keep that blood within a stable pH range. Same is true when the hydrogen ions are depleted. They're going to release those hydrogen ions to get that pH back to where it needs to be. All right, this is just kind of a visual to show you once again those hydrogen ions, important role in pH. We can see more hydrogen ions, it's going to be acidic. Fewer hydrogen ions, it's going to be basic. I'm not going to spend too much time on this slide. The last thing I really wanted to mention was the pH scale, and that it's logarithmic. All right, so it's not just that pure water is 7 and say urine is 6. All right, urine is actually 10 times more acidic than pure water. Same with if we had a solution that was at a pH of 5, that's 10 times more acidic than a pH of 6, 100 times more acidic than a pH of 7. All right, so there's a vast scale of pHs. All right, and the reason it's logarithmic is because there's so many ranges of pH that we can have in biology that we had to condense it down to make it kind of simpler to understand per se. All right, and pH affects a lot of things, and we're going to see this come up time and time again as we go on to different things such as chemical reactions and enzymes. The pH is going to affect the shape of molecules, the rates of chemical reactions, the ability of molecules to bind or dissolve. pH really affects a lot, which is why we have those buffers in our system to try to keep the pH in our bodies kind of in that optimal range. All right, lastly, I had a question on Friday. Where do electrons come from? All right, so we talked about ions, how they, if they gain an electron or lose an electron, they can become an ion, or they can have a charge associated to them. All right, so I did some digging, and essentially, electrons are just free roaming until they are, quote unquote, captured by another atom. All right, so we can have free roaming electrons, and we actually use this to our advantage in terms of electricity, or I don't really understand how electron beams work, but that's how the old TVs worked, by using electron beams, so just free roaming electrons. We can artificially release electrons from atoms if we heat up a substance really high, causes them to exit that outer orbital. So electrons, they can be free roaming. That's where they come from. I think the ultimate answer to that is they came from the Big Bang. So if we really want to go big here, that's kind of where they were created per se. But essentially, they're just free roaming. All right, any questions about what we just covered? I know I kind of went fast. All right, here's some additional resources for you guys on this topic. I highly recommend this podcast. It kind of intersects arts and science together. It's pretty cool. Don't have to watch these. I'm not going to test you on these additional resources, but they're interesting. They might help you understand things a little bit better or give you a new perspective. And then hopefully you guys all watch these videos that I had you guys watch as prep for today, because we're going to move on. All right, so I'll give you guys a chance to change PowerPoints or switch it up in your computer. I'm going to pass around the properties of water worksheet. So basically, I am giving you a property of water. I am asking you to describe that property and then what the biological consequence of that property might be. All right, now that biological consequence, don't get hung up on little tiny stuff like we've been talking about. Think about the ecosystem as a whole. Don't think about it just in terms of molecules. All right, so broaden your range and it'll be a much easier answer to answer here. Question to answer. I am going to, the easiest way to start is over here, so I can help you guys easier. 7. And again, this is worth 3 points. Bring it next week, not next week, next lecture on Wednesday. And if you have any questions, we can go over it at that time. I'm just going to start on this end and we'll make our way to the middle there. All right, so we kind of got over the nitty-gritty of the small molecular workings of atoms here. We're going to start getting a little bigger in scale, not by much, but a little bigger here. And we're going to talk about the building blocks of life. All right, these are the key concepts. Not going to go over them just for your reference here. But we should have watched that video on carbon. I think there's actually 2 videos. And so I'm not going to go too in depth of carbon here, but I want to review. Yes. One more. Let's see. Do we have an extra one? Awesome. We can make that come over here. That would be awesome. Everybody else got one though? All right. Awesome. All right. So carbon. How many bonds can it form? Potentially. 4. Yes. And why is this? 4 valence electrons, yes. It has 4 valence electrons. It can form 4 bonds because it wants to get that outer orbital full. Now, is this configuration always going to be the same? No. What are some examples of ways that these carbon bonds can be formed and configured into three-dimensional structures? Rings, yes. Anything else?
Flat sheets.
Flat sheets, yep, just kind of linear. Another big one out there. Tetrahedrons, yep. Going back to kind of our three-dimensional structure and rings. Yes, that is far beyond what I know or expect you guys to know, but that is awesome. The last one I'm thinking of is much simpler, just branching. All right, so it's going to branch. It's not going to be just simply linear. All right. So carbon. All right, carbon can form polar or nonpolar bonds. These bonds are going to be very stable because if you think about carbon, carbon is a small molecule compared to those other elements on the periodic table. All right, because they're small, they can fit together tighter. Those bonds are going to be closer to together, so it's going to be very stable. Now, when we talk about carbon, we can talk about hydrocarbons. Hydrocarbons are nonpolar, all right, and these are going to exist of purely carbon to carbon or carbon to hydrogen bonds. All right, so those electronegativities are the same, so there's going to be no polarity associated with these. Because they're nonpolar, all right, they're going to be very insoluble. They are not going to dissolve in water. Now, oxygen and nitrogen, if we enter them into the equation here and form bonds with carbon, they're going to form polar bonds because of those differing electronegativities. Now, they're going to be hydrophilic or water-loving, which means these bonds are going to dissolve in water. Now, as we kind of touched on with those micelles, all right, we can have one molecule that has a polar and a nonpolar region. A good example is this acid up here. All right, we'll have only carbon to hydrogen or carbon to carbon bonds on this end. So this is our non-polar region. Likewise, we have oxygen over on this end, different electronegativities. So it's going to create a polar end to this molecule. Now through this up here, just kind of going back to what we talked about in our question here, we can have many different shapes depending on how carbon is arranged. All right, so right here we're looking at only hydrocarbons because we only have carbon and hydrogen. And depending on how they're arranged can totally change the shape of a molecule. So much so that even if it has the same chemical formula, if it's arranged differently, such as in the branching or the double bonds example here, it's a totally different molecule. All right, so placement is very crucial. Now, if they do have the same formula, but they look different, have different arrangements, we call those isomers. All right, so I went over carbon pretty fast because you guys should have a solid understanding of it. We're going to move on to functional groups. All right, so most organic molecules are going to contain functional groups. All right, and carbon is a part of many of these functional groups, which is why we started with talking about carbon. But functional groups are going to be groups of atoms that have specific chemical features and are always going to function the same way. All right, so they're groups of atoms. They can be on many different molecules, right? So it doesn't always have to be attached to the same molecule. They can be attached to different molecules, but it's always going to be the same group of atoms that are going to be attached, and they're always going to behave the same way. All right, so for example, the amino group here, NH2, it's going to act as a base no matter what molecule it's attached to. All right, it's always going to act as a base. Now, I threw up a table with some of our biologically important functional groups. I don't expect you guys to necessarily memorize these, all right, but I want you to know that they're out there, they exist. We will be talking about these further along, so I want you guys to kind of have an overview of this. Now, this is not a complete list of the functional groups. The complete table is in your textbook if you want to learn more there. All right, so beyond functional groups, all right, as we start adding on to our knowledge and start building these molecules we're talking about here, we're going to move on to just biological molecules, all right? They can be very, very large. So far we've been talking about a singular atom or very small molecules. These small molecules we could also say were monomers, all right? They're just a small molecule. Now if we put these monomers together, we'll get polymers, all right? So we're adding in complexity here. So the same molecule strung together, we're going to end up with the polymer. Now if we get a very, very, very large polymer, it becomes so large that we start to refer to it as a macromolecule. So how do we get these macromolecules? How exactly do these monomers turn into polymers, turn into macromolecules? All right, it all has to do with chemical reactions. All right, so we're going to form and break macromolecules, do these chemical reactions. So in this image here, you can see we have our growing polymer. All right, this is just a generalized polymer made up of many different monomers. And on the end here, we're adding more monomers to our polymer. We're growing our polymer out. And this is done via a polymerization. All right, so this is the bonding together of monomers. So once we get a very large polymer, we're going to get a macromolecule. All right, so this polymerization, we're bonding together those monomers, and we're doing that via dehydration reactions. All right, so we're going to very broadly go over these reactions here, but essentially what is happening is we're bringing two monomers together, and we're taking away a molecule of water, and in the process of taking away that water, we are bonding those two monomers together. All right, so in this image here, we have our monomer here, and we have a second monomer here. On the ends, we have two hydrogens and one oxygen. All right, they're going to combine to form H2O, and in turn, link those two monomers together. All right, so this is polymerization right here via these dehydration reactions. Now we can do this over and over and over again to get larger and larger polymers. All right, Like it says here, a polymer can consist of thousands of monomers. And I should mention it's also catalyzed by enzymes, both dehydration reactions and our next reaction, hydrolysis here. Enzyme is just a protein that's going to help speed up a reaction. We'll talk more about those in the upcoming weeks here. But enzymes play an important role in making this all come together. Now, just like dehydration, we can have hydrolysis reactions where we're actually breaking apart the polymers. All right, so this is important. A lot of plants, they will store energy in large polymers inside these storage vesicles. And when it comes time to actually use them, they need to break these polymers down in order to transport them throughout the plant. All right, so we also need to be able to take apart these polymers. And the opposite reaction is true. We are just bringing water into the reaction, all right, and we're going to break apart a monomer. All right, any questions about dehydration reactions, polymers, monomers? All right, we're going to continue moving on here. I think we're actually making decent time. So there's 4 main types of organic molecules that we're going to talk about when we talk about the building blocks. All right, and today we're only going to focus on 2 of those, which is carbohydrates and lipids. All right, carbohydrates. Carbohydrates, they're going to be composed of carbon, hydrogen, oxygen. All right, and they can range from very small sugar molecules all the way up to what we call polysaccharides. All right, so let's kind of start at the small sugar molecule end of carbohydrates here. So a single sugar monomer is what is called a monosaccharide. All right, so down here we can see we have a monosaccharide of glucose and a monosaccharide of fructose. All right, they have the same chemical formula. So does anybody remember the term for what that makes this? Isomer, yes. So these 2 are isomers. Same chemical formula, but slightly different arrangement makes them a different molecule. All right, we also have, I should back up a little bit. All right, so glucose, fructose. Right here we have them kind of laid out in these linear forms. They can exist linearly, but most common they're going to exist in these carbon rings. All right, so these are example of monomers, galactose, glucose, fructose. All right, pretty common sugars. Now for monosaccharides, we can start building bigger molecules. All right, so monosaccharides, they're extremely important in fueling cellular work. They help to create ATP. They're used to manufacture other organic molecules. So really they are a building block to the synthesis of a lot of other molecules in our body. Now if we put 2 of these monosaccharides together, we get a disaccharide. All right, so di meaning 2, disaccharide. Now these 2 sugars, once they're put together, they're going to be put together via a dehydration reaction, just like we just learned about here. But the process of putting 2 sugars together via dehydration reaction is going to make what's called a glycosidic bond. All right, so a specific name for that bond when it occurs between 2 sugars. Now if we go even further, all right, so we got the monomer, we got the polymer, or the monosaccharide, and we have a disaccharide, we can get polysaccharides. All right, so macromolecules, they're polymers of thousands of monosaccharides. They are very, very common in our biology and the biology around us, and they play really important functions. All right, I'm actually going to have you guys investigate these functions on your own. So as a group, all right, I want you guys to think about why the following polysaccharides are biologically important. So I listed 4 there. I want you guys to take out your phone, go on Google, whatever you need to do here, textbook, figure out why are they biologically important. It's also a good time to ask any questions that you don't want to ask in a big group.
I would just say this one. You guys want to go over? Ready? They go over me.
Okay, how's it coming? Quieter in here. Does that mean we're all kind of wrapping up? Anyone need more time? All right, let's come together and discuss. All right, so starches. Why are they important? Okay, where could starches be found?
In many foods like potatoes and chips.
In many foods, yes.
Bread.
And what do they do?
Store energy for plants.
They store energy, yes. So think about a potato. Think about the tuber that's underground. That's an energy storage mechanism for the plant. They store energy. All right, what about glycogen?
It
Yes. Yeah, so now we're talking about more animals now. Glycogen is also an energy storage uh, polymer here that stores energy in animal tissues. What about cellulose? What is the function of cellulose? Makes up the plant wall, yes. And what does it do for the plant wall? Stabilizes it, creates a structure, yes, a very rigid structure. And what about chitin?
It's a key component in like the exoskeleton for arthropods.
Yeah, so think of crustaceans and like let's see, a lobster, for example. They have chitin in their shells. And what does this do for these arthropods? What was that? Protects them. Yeah, provides protection for them. So, polysaccharides, very diverse in what they can do on, plants versus animals. Some things might have the same functions, but very biologically important. And this is only touching on 4 of them. All right, there's a ton of them out there. All right, any questions about carbohydrates? All right, we're going to move right on into lipids then. All right, so lipids are going to be insoluble in water. This is a key component about lipids, is that they are hydrophobic. All right, so hydrophobic, water-fearing. Lipids are going to mainly have carbon, hydrogen, and oxygen, and are going to be linked by those non-polar covalent bonds. Again, non-polar covalent bonds means they're going to be hydrophobic. All right, so these are kind of key components of lipids in general. Now, lipids, they're going to vary in structure and function, just like everything else we have seen. And we're going to talk about these 3 main types and kind of touch on a 4th. All right, so we're going to talk about fats, phospholipids, and steroids, and just touch on waxes a little bit. So lipids are different in other biological molecules, especially from carbohydrates that we just talked about here, in that they're not very big. All right, they're not these huge polymers that we had kind of been talking about with like starches or cellulose. They're actually very, very small. I don't want to say small as in tiny, but they're a lot smaller than, say, a starch. They're not going to be built from monomers. All right, so that's another key difference here. We're not talking about monomers anymore. So we're going to jump right on in and talk about fats. All right, fats, also known as triglycerides. All right, fats are made by bonding these 2 key molecules together, glycerol and fatty acids. All right, so glycerol, we have our glycerol molecule down here on the bottom, and we have our fatty acids. Now, fats will bind 3 fatty acids to a glycerol molecule, and as we have seen so often now, this is done via a hydrolysis reaction. All right, so we're taking away water. Did I write that? No, I totally said that wrong. It's joined by dehydration and broken apart by hydrolysis, just like we've seen in the past. So let's see here. So on here we have our glycerol. All right, we have these hydrogen atoms on the end here. We also have these fatty acids. Now, if you remember back to those functional groups, I don't expect you guys to know them, but they're going to come up in our terminology. This is our carboxyl group. So this is one of those functional groups. They're going to be found on the ends of fatty acids. And they're going to help join the fatty acid to the glycerol via the dehydration reaction. All right, so we're taking away water and joining these molecules together. All right, this creates a new bond. We call this an ester bond. And creates a fat or a triglyceride. Now, a big component of these triglycerides is they help to store energy. All right, so many molecules help to store energy. Fats are another one of them. Now, fatty acids are going to differ from each other based on their length and the presence of double bonds. So let's look at saturated fatty acids. All right, you've probably heard this term before. Saturated fatty acids are going to have the maximum amount of hydrogen that you can possibly have because they don't have any double bonds. All right, there's no double bonds present, meaning every bond that can be formed with carbon and hydrogen is present. So we're packing in those hydrogens. All right, kind of a key characteristic of these saturated fatty acids, they can solidify at room temperature. It takes a lot of energy to melt them. Commonly found in like animal fats. Let's see, if you eat meat and fry up a burger and then you put it in the fridge for later, maybe you've seen some of that fat solidify in the fridge. All right, so that's the kind of fats we're talking about here, animal fats. The other type is an unsaturated fatty acid. All right, so basically in an unsaturated fatty acid, you're going to have at least one double bond. And because you have one double bond, that means that there's one bond that could have had hydrogen that doesn't because it's double bonded to, say, another carbon. All right, so we're missing at least one hydrogen that could be in our fatty acid. This means it cannot solidify at room temperature, and these are most commonly found in like olive oil, corn oil, vegetable oil, different types of plant oils. So if we look at our image over here, right, we have our glycerol molecule, and we have our 3 fatty acids. Now, by themselves, the first 2 fatty acids, they would be a saturated fatty acid because they don't have any double bonds. But because they're grouped with that one fatty acid on the end that has that double bond, we consider the whole molecule an unsaturated fatty acid, right? It has the potential to have one more hydrogen in it, but instead it has this double bond. Any questions about those fatty acids? All right, I think they're pretty easy to understand here. We're going to touch on steroids. So steroids is another class of lipids. And this is where the carbon is going to be in 4 carbon rings. All right, that's very characteristic of a steroid to have 4 carbon rings. A very common one is cholesterol, which we can see on the side here. Now, depending on which functional group might be attached to these carbon rings, are going to give them very different functions. All right, I just started with cholesterol because it's very common, and it can be the backbone to build a lot of other steroids off of. Other common steroids are sex hormones, such as testosterone or estradiol. Testosterone in particular is part of what we might commonly think in mainstream as a steroid. Right? An anabolic steroid is how we get these big bodybuilders, which I cannot see how this would be comfortable, but to each their own. So these steroids are going to be a variant of testosterone that basically help you build muscle and bone mass. All right? A lot of people will take them if they're bodybuilders. Maybe they'll misuse them if they're an athlete. But they can also help treat certain diseases such as anemia or diseases that might destroy body tissue. All right, so we can take a steroid to help those individuals build that body tissue back up to where they should be. They can also have very violent or very, what's the word I'm looking for? Nasty side effects. All right, so anything from, mood swings to cancers. All right, there's a lot of side effects that can be associated with these steroids. All right, didn't want to touch too much on the steroids here, but I wanted to mention them. We're going to move on to phospholipids, which is super, super important in biology. I think this might be the most important lipid, but that's just my opinion. So phospholipids are very similar to fats in that they are attached to a glycerol and they have a fatty acid. Now the important difference is that they only have 2 fatty acids, and instead of having a 3rd fatty acid, they have a phosphate group. All right, so they have a phosphate group, glycerol, 2 fatty acids. Another thing to note is that they also have this positively charged nitrogen region. All right, so if we hear the word charged, that immediately makes us think that it's polar. All right, so this is an example of one of those molecules that have polar and nonpolar regions. Very characteristic of a phospholipid, and this makes them a major component in cell membranes. All right, so just kind of a different image to reiterate this. We have our charged nitrogen area. We have a phosphate group. We got our glycerol and our fatty acids attached. Now, because one side is polar, one side is not non-polar, it kind of acts similar to those micelles we were learning about, but instead of forming a circular ring, they form these bilayers. All right, so cell membranes, they are formed out of phospholipid bilayers. All right, and they're arranged so that the nonpolar regions are towards each other because they are hydrophobic. They don't want to interact with any water that's around them. And then the polar regions are on the outsides. All right, I'm sure you guys have seen this image before in general biology classes. Very characteristic of a phospholipid to form these bilayers. Now, these bilayers have different ranges of permeability. We're not going to address this too much right now, but I wanted to introduce the concept that because of these bilayers, all right, and how they are packed together, small nonpolar molecules are able to easily pass through them, right? So they can pass through these membranes without the use of, say, a transport or a protein or anything special. No different channels. They just can pass right on through. But they also exclude larger or charged molecules, such as ions or large polysaccharides, for example. All right, they'll exclude the larger things, but allow the smaller things through. Is kind of the general gist of what I want you guys to get out of this for now. And finally, we have waxes. I think waxes are pretty cool. All right, they're kind of a complex liquid that plants and animals use in many different ways. They're very nonpolar, all right? And this is very characteristic of a wax to exclude water. And so kind of an example would be a leaf here. You can see how these water molecules are kind of on droplets on top of one each other. Let's see, they're forming droplets due to those properties of water, cohesion and adhesion. So that's another terminology we can put in there. But it's on this waxy surface, all right? So it's just kind of rolling off the leaves. It's the waxy surface is protecting these organisms from water loss in terms of plants, or helping to build these structural elements in terms of a honeycomb. All right, one last group question for y'all. All right, so we know that oil and water does not mix. All right, but we also know there's some products such as mayonnaise where they contain both water and oil, and it's all mixed together. All right, how does this work? All right, so I want you guys to kind of dive into that and Google how does an emulsifier work. All right, so a type of emulsifier would be a lectin, which is a molecule found in egg yolk. I want to know how does that promote water and oil actually mixing together. So again, use your phones, use your computer, Google how do emulsifiers work together as a group, and then we'll come back together. So key component of working in a group, be talking to one another, bouncing ideas off of each other.
It's like dumb down answer.
All right, starting to quiet down. We only have like a minute left here. So, who has an answer for me? Who would like to tell me how this works?
Like the outside is polar, so it's able to like attract water and other polar particles, and the inner is non-polar, so it kind of acts like almost like a deterrent, but instead of using like food stuff, so it's able to both attract like fatty parts and also like the water base.
Yeah, that's exactly it. It's just kind of connecting those two components together, working again very similar to how a mycel would work. All right. Additional resources for you guys, videos that I think are nice and helpful. They're nice and visual, which, you know, I know for me, I'm a visual learner, so this helps me a lot. And then prep for Wednesday. All right, again, I'm going to have you guys watch a couple videos. One video, you only have to watch up to like minute 3. The rest of it, you can keep watching, but it's totally extra. And then bring that completed H2O assignment. Yeah.